Hello. Thanks to the organizers for allowing Freenome to present some of our latest results on there in terms of technology. My name is Jimmy Lin. I'm the Chief Scientific Officer of Freenome. I will be presenting two talks here at the conference. The first talk is talking about our technology. The title of the talk is Blood-Based Multiomics Technology for Lung Cancer Screening. For those who don't know Freenome is an early cancer diagnostic company based in the San Francisco Bay Area. Sorry I couldn't be there with you in person there. We are a company that's trying to be able to catch cancers early. And our approach is be able to take indications that have a large unmet need, and we feel like we are able to make a difference there. Obviously, this is focusing on lung cancer, and we think that lung cancer screening is important. Early detection makes an impact. If you catch it in early stage, the survival is really high, over 65%, but if it's metastatic, it's very low. So it's important to be able to catch these lung cancer people who are eligible for lung cancer screening. But the sad reality is that there's very low adherence. 10 to 15% of people who are eligible get screened there, even though that there is very good technology. So we're building a blood test that's very similar in patients who are initially sort of triaged for low-dose CT there. And the intended use is over 50 years, over 20 pack year smoking history there. So that's what we're trying to build. Our approach is we build on a unique approach here at Freenome. It's because we think that the way to lung cancer screening is not actually directly through lung cancer screening. We actually think that it's through other methods. This is a paper published in JAMA a few years back there. What's interesting is you're looking at panel B here. It's the use of preventive health care among US adults who are eligible for lung cancer screening, so LCS, and as well as ineligible. Let's just look at this panel here. For the patients who are eligible for lung cancer screening, their screening for colon cancer screening is a little bit lower than the ones that are ineligible, so they're a little bit lower, but in comparison, it's still pretty high. It's still over 60%. So that even though the patients eligible for lung cancer screening have low rates in terms of being screened for lung cancer, which is shown here, but they actually participate in other kinds of cancer screening, whether here shown for colorectal cancer screening or breast cancer screening. So we think that this is an opportunity to be able to leverage, if they are doing other types of screening, to add this on top of that, to be able to do that. Freenome has already been working on a colorectal cancer screening test there. And this test, when added upon the existing colon test, when you add the lung cancer, hopefully will really sort of make the adoption really easy. That the patients do not have to collect any extra blood, or it'll be just part of their colon screening efforts, and they could be added on top of that. So that's our sort of unique approach to that. We have a test right now that's sort of under FDA review called Simple Screen CRC. It's for all patients who are eligible for colon cancer screening over age of 45 there. So this is a large area, large intended use population, and many patients who are eligible for lung cancer screening actually fall into that. So that overlap is an opportunity for having higher adoption and adherence to lung cancer there. We published our blood cancer screening test in colorectal cancer last year. We did a trial called PreEM-CRC that enrolled close to 50,000 patients and ultimately analyzed about 27,000 patients there. And it's one of the best-in-class blood tests that's out there. Right now, we are working with the FDA to bring this available to market. Once available, then we are working to add the lung test on top of that, and hopefully to be able to screen for many patients for lung cancer who otherwise would not have been screened. Our test right now, even though that the first test is designed for lung cancer, it actually has markers for 10 other cancer types, including lung. So again, the same test can be used, added on pretty easily in that sense. With their consent and with the order, additional computation analysis can be done on it. So without any additional sort of blood draw, we think that's the way to be able to really increase the adherence there. So the technology that we start off with to be able to find these markers, and we do this for all our cancer types, starts off with multiomics there. Because we don't know what's the actual sort of data that's going to be important for cancer screening for every cancer type. So we look at all the ones that are available to us, DNA, protein, RNA, other analytes there, and we've built this multiomics platform within the last 10 years. Then we apply it to different sets of cancer samples. So we did it earlier on with colorectal cancer, and now we're doing it with lung cancer. And you added that with real world data and feedback loop. This becomes very, very powerful in terms of developing cancer screening tests there. For sample, this is what it looks like. You have a sample that comes in when we do our discovery. The DNA can be used for genome sequencing or sort of methylation analysis. The protein can be used, and we've used them for mass spec, for immunoassays. We also can do methods such as done it in RNAs, autoantibodies, and others there. So this allows a very sort of broad discovery effort there. Let me talk a little bit about one technology in particular, methylation, which is actually one of our most powerful technologies that we've developed at Freenome. Methylation detects DNA called cell-free DNA as dying cells. Cells die for apoptosis and necrosis. They release DNA into it. But tumors also release DNA called ctDNA, circulating tumor cells. And the goal here is for us to be able to detect both the cfDNA and ctDNA, be able to distinguish and to be able to find those signals early. And then we do it here through the method methylation, DNA methylation method there through next generation sequencing. What we do here is not only sort of capture the fragment level in terms of, some fragments are methylated versus not, but we actually look at down to the base level. We can tell is whether a fragment is methylated at 3 positions versus 1 position there. Down to this base level allows us to have very, very exquisite LOD and sensitivity there. We've shown this in colon cancer, and we're continuing to start showing this in lung cancer as well. So this is unique technology that's developed to give you a sense of the technology that enables some of this in terms of our multi-omics effort there. And then with all this data coming out, a lot of computation analysis is required. This is one example of the type of analysis that we did. This is specifically looking at sort of using deep learning, machine learning methods there. In fact, this is being presented this week at the NVIDIA conference at GTC. They're using a specific method called multi-instance learning to be able to find, again, the needle in this haystack signal molecule there. So using proprietary and uniquely developed technology there, and also machine learning and computational methods, we can then start looking at these samples in many different contexts there. So what does it look like in lung cancer? Due to a short talk, I'll just give you a quick preview here. We apply this to a set of samples there, 418 samples, and as many of you know, lung cancer has many different subtypes. So we've applied them to make sure that it has performance in adenos and squam and other cancer types as well, with sort of matched negatives. And you can see here, looking at blue, which is the methylation signal. The orange here is the protein signal, and then you can see them when you sort of add them together is the green signal. Here's the true positive rate there. You can see here the true positive rate here at 75% spec is 80% sensitivity there. We're continuing to make improvements on this there. And in fact, a little later next month, we will be presenting at AACR using our latest multi-omics test, which combines methylation and protein. If any of you are there, come visit our poster there at AACR. And we're continuing to build this. So we are going to be launching this test there as an LDT here this year. We're working on the AV-CV as we speak. We'll be launching this at the second half of this year. We are also enrolling for a PROACT test for our FDA trial there for our PMA, and that's sort of ongoing as well for ultimately be able to have this rolled out not only as an LDT, but also with the FDA as well. So we're currently enrolling and pretty excited as we continue to develop a test, that hopefully a best-in-class test there, and be able to help many patients in the future. That was a quick whirlwind tour in 10 minutes of our lung cancer screening efforts and some of our technology. Hopefully, this gives you a glimpse here. This is a photo day for us, a place of us in the Bay Area. So if you're ever in the Bay Area, definitely come and visit us. So I can there be in person and answer questions, but if you have any questions, my email here is below, jimmy.lin@freenom.com. Hope you have a wonderful conference.